Hello everyone, my name is Hanh Nguyen and I will be presenting about my PhD project, which is called Wildlife and Microbiomes in the City. So, as introduced by Emily before, the world as we know it is defined by microbiomes. Microbes are everywhere. Microbiomes are microbes and they're relatively well-defined habitats. Microbes are everywhere, so microbiomes are also everywhere. And they have been identified as very important to maintain the health of animals, either directly through their many contributions to health of the ecosystem, or directly through training the immune system of hosts, and their involvement in brain function and energy storage and usage. So urban lands are expected to increase threefold between 2000 and 2030, making it the fastest growing ecosystem in the world. So with urban lands increasing exponentially, the focus is now on the microbiome of cities. But it is a complicated picture because there are multiple habitats within cities. And studies have yielded different results as well. So we have both found cities to have less diverse and lower abundance of microbes, but we have also found cities to host thousands of taxa of microbes not matching any previously known organism. So there's an ambiguity in the way that urbanization might have affected the microbiomes and the origins of their components in cities, and making it hard to draw conclusions about the characteristics of microbiomes in the cities and how that the impact of urbanization on the microbiomes and health. So within cities, we have green spaces, we build green spaces for the benefits of human health. Green spaces include many different facilities ranging from forests, parks to garden yards and remnant patches of native vegetation. These green spaces, beside human health benefit, also provide important refuge and habitats to animals. Therefore, the planning and management of these green spaces would also play an important part in protecting biodiversity. In Melbourne, there's a study that have found green space management strategies aimed for improving biodiversity, improving urban biodiversity, have a positive effect on the assemblages of bats and birds in the area. So this effect on animal aggregations and population at the site might also subsequently affect the soil microbiome of the area, as animals frequently interact with soil, either through eating the vegetation that shapes the soil microbiomes, or through various direct activities like scratching or digging, or through defecation. So the aim of my project is to investigate how green space management intensity might influence the effect of city living on microbiome of wildlife and how it changes the way animals might infect the soil microbiomes of their habitats. And since it's kind of small, the text in the middle is me explaining to my friend about my PhD project and them calling me Dr. Dirt. So this is my field design. So I aim to carry out this experiment on 4 different cities: Melbourne, Central Melbourne, Hobart in Tasmania, Central Melbourne, Sunbury, and Ballarat in Victoria. Within each city, I will select 3 grassy areas so we target grassland especially for this project to avoid the influence that bushes and trees might have on the soil microbiome. And these grasslands area might be riparian parks, big parks, and small parks. So within each of these parks, I will select 2 areas, one with high maintenance intensity and one with low maintenance intensity. And within each area, I will set up 4 plots. So if you can see, there will be fence plots with local soil, unfenced plot with local soil, fence plot with sterilized soil, and unfenced plot with unsterilized soil. Wait, hang on, local soil. So first, before any soil collection can happen, we will carry out a population survey at the area, an animal population survey at the area to investigate the animal population. We'll be doing this through bird spotting and scat survey. The scat sample will also be collected, as I am also interested in finding out how city living might influence the microbiomes of animals. So, how did the soil survey happen? Well, we intended to carry out the soil survey throughout a 6 months period. So I would be collecting one soil sample at 0 month, and then 3 months afterward, and then 3 months after that time. We do this by comparing between sterilized and unsterilized plot of each kind, fenced and unfenced. We hope to be able to identify seasonal variation and tease out the animal influence changes in the soil microbiome and investigate how the microbes might colonize the soil. Between fenced and unfenced plots, we'll be able to investigate the direct effect of animals on the soil microbiomes. Bless you. So what is the purpose of this? Well, overall, I want to understand the effect of management intensity on soil and wildlife microbiomes, the relationship between animals and soil microbiome, and the implications of those interactions for human and non-human animals' health. Thus, the result we might be able to inform conservation and restoration planning and the creation for a healthy urban ecosystem. So the progress so far of the year of lying on the couch being anxious about the state of the world, I have published one paper on a journal called Trends in Ecology and Evolution. I've also been granted a Holsworth grant that will go toward the fieldwork portion of this project, which is, why I'm swimming in Scrooge McDuck money. And I am in the progress of contacting councils and partners for permits and setup of the fieldwork. Oh, any questions?